Hello everyone, hope you're all safe. So in our previous video, I have given an introduction regarding enzyme inhibitors and its various types. In this video, we will be dealing with the different types of enzyme inhibitors in detail, and also we will be discussing about regulation of enzymes. So let's start our class. Enzyme inhibitors. So there are two types, reversible inhibitors and irreversible inhibitors. Also the regulation of enzymes. So enzyme inhibition. Enzyme inhibition means these are molecules that bind to the enzyme and they will interfere its activity. They can also prevent the formation of enzyme substrate complex and prevent the breakdown of enzyme and product. And they use chemicals that reduces the rate of these enzymatic reactions. So first one, that is reversible inhibitors. So reversible inhibitors means they'll bind to enzyme through non-covalent bond. Using non-covalent bond, they are binding to the enzymes. So here the enzyme is not permanently inhibited or damaged. Here inhibition can be reversed when inhibitor is removed. So there are three types: competitive, non-competitive and uncompetitive. First one, competitive inhibitor. In competitive inhibitor, they'll binds to active site and results in inhibitor enzyme complexes. So here there will be competition between enzyme and substrate, but they will reduces the number of enzyme molecules for substrate molecules to bind. So as a result, less catalysis will take place and rate of the reaction will also decreases, because these both molecules are competing for a same active site, and this reaction depends upon the concentration of this substrate and inhibitors. See, this is a competitive inhibition example and this is active site. So this is inhibitor and this is substrate, and here these both are competing for this active site. And one of the example for competitive inhibition, this is succinate. Succinate will undergo reaction with succinate dehydrogenase to form fumarate. Also they can undergo with malonate to form fumarate. So here malonate is the competitive inhibitor for succinate dehydrogenase. Non-competitive inhibitor. In non-competitive inhibitor, here they will not compete with substrate for the active site. They will bind at any part of the enzyme. So this is sometimes called allosteric inhibition. As a result of this inhibition, there will be change in the shape of active site, because the substrate will not fix for long time. So this type of inhibition can widely seen in the biochemical pathways such as glycolysis and Krebs cycles. See, this is the example for non-competitive inhibition. Here cyanide will compete with iron for the cytochrome oxidase. So this is substrate and this is enzyme. So there will be no competition between this. So this will bind to any region of the active site. So in non-competitive inhibition, there will be change in the shape of enzyme. So it cannot bind to the substrate. In competitive inhibition, they will interfere with active site. This is the active site. So the substrate cannot bind. Uncompetitive inhibitor. So this will binds only to substrate enzyme complexes and it is created upon the substrate binding. It is very rare class of inhibition. Example is tetramethyl sulfoxide and 3-butyl thioline 1-oxide. These are the examples and these are the inhibitors for the alcohol dehydrogenase. Irreversible inhibitors. Irreversible inhibitors will bind to the enzyme through covalent bond. In reversible, non-covalent bond, and for irreversible they are covalent bond, and they causes chemical changes to the active sites of the enzyme, so they cannot be reversed. And the main function of these inhibitors is we are modifying the amino acid residues that is required for the enzymatic activity. And they also contain functional groups such as aldehydes, alkenes, phenyl sulfonates, etc. And there will be electrophilic groups. These electrophilic groups will react with this amino acid side chains to form covalent adducts. And these amino acid components, these are the residues. They will contain nucleophilic side chains such as hydroxyl, sulfahydryl group, etc. And these are the examples of the irreversible inhibitors, that is substrate analogues, affinity labels, suicidal inhibitors, etc. Regulation of enzymes. There are two types, that is enzyme induction and enzyme repression. Two types of regulation of enzymes. Enzyme induction. Enzyme induction means there will be increase in the rate of synthesis of enzymes because of the presence of inducer. So with the help of inducer, we will increases the rate of reaction of enzymes. In other words, we can say they will depresses the gene that is responsible for the synthesis. Enzyme induction, it is a process where the enzyme is manufactured with the presence of a specific molecule. Specific molecule means inducer only. That is, with the help of inducer only, the enzyme will get manufactured. An inducer molecule that combines with the another molecule, so that is called a repressor. And this inducer will bind to the repressor. They will block the function of the repressor, and that is bind to the another specific site, that is called operator. And this operator site, another molecule get binded, that is known as ribonucleic acid, that is RNA polymerase. Again, the binding takes places and they will begins the transcription process. So RNA will get produced, messenger RNA is produced. They will act as a template for the production of protein. This transcription process we can detailedly studied in the coming chapters. So this inducer and the repressor binding keeps the repressor from preventing the transcription. And gene coding of this enzyme is also transcripted. In bacteria, that is lactose, lac-operon, it is a very good example that operate based on this induction. Next is enzyme repression. In enzyme repression, they will prevent the manufacture of enzyme. They also operated by feedback inhibition. this repressor will bind with another molecule and they are able to block the operation of this operator. And this blockage can occur. The repressor will outcompete with this polymerase for binding on the operator. Alternatively, this repressor will get directly bind with this polymerase, thereby there will be change in the shape of this polymerase. So they will bind to the operator region, and as a result the blockage of transcription on the particular gene. So this is enzyme repression. So that's for today. Thank you.